Do you know anyone with diabetes who is taking insulin? If so, then they are reaping the benefits of DNA technology. The human insulin protein hormone is produced and purified from bacteria to treat diabetes in humans. While DNA technology is a relatively new field, it has more applications than you probably realize, from agriculture to medicine to forensics. DNA technology involves the manipulation of genes to achieve a practical purpose or goal to benefit humanity. It's used in research and many other fields, including medicine, agriculture, and forensics. In medicine, genetic testing, gene therapy, and stem cell therapy can be used to diagnose, treat, and cure disorders. In agriculture, genetically modified organisms are generated to yield desirable characteristics. And in forensics, the process of DNA profiling is used to convict or exonerate suspects. DNA technology involves moving genes from cell to cell or from one organism to another. Different tools are used to manipulate everything from short stretches of DNA like genes to all of the DNA in a cell. I'm sure you've heard of different fields of engineering like mechanical or chemical engineering, but did you know there's a field of engineering called genetic engineering that involves the modification of genetic material in which genes may be deleted, added, or changed? This is used in research, agriculture, and medicine to generate what's called recombinant DNA. Recombinant DNA is DNA that contains genes from different organisms. Recombinant refers to combining genes from different organisms into one piece of DNA. Recombinant DNA is produced in simpler organisms like bacteria, viruses, or yeast, and then transferred into higher organisms including plants and animals. This generates transgenic or genetically modified organisms, which are the organisms that express this recombinant DNA. Many transgenic organisms have been generated, including bacteria, yeast, plants, and animals. Bacteria are prokaryotes, which are single-celled organisms that lack most of the organelles or specialized structures that eukaryotes have. Yeast are single-celled eukaryotes, so they have these specialized organelles, but they're simple organisms containing only one cell. Plants and animals, which are multicellular eukaryotes, are more challenging to generate. In the next few slides, we're going to discuss how all of these transgenic organisms have been generated and the applications that they're used in. First, we're going to discuss how transgenic bacteria and yeast are generated. First, a restriction enzyme is used to cut the gene of interest out of the chromosome in which it resides, and also to cut a plasmid, which is a circular DNA molecule found in bacteria. This opens up the plasmid to allow the gene of interest to be inserted. Restriction enzymes cut DNA at a specific sequence to yield single-stranded overhangs of DNA. When the gene is mixed with the plasmid, these complementary overhangs will bind. Keep in mind that single-stranded DNA will bind to other single-stranded DNA if it's complementary to one another. The recombinant plasmid with the gene of interest is now moved into a recipient cell using the process of transformation. Transformation involves bacteria or yeast taking up DNA from the environment. You may wonder why the gene can't be put in the bacteria or yeast directly, and why it has to be put in a plasmid. A plasmid essentially provides a shuttle to get the gene of interest into the bacteria or yeast, since these organisms readily take up this type of DNA from the environment. This diagram shows the steps involved in generating transgenic bacteria. The gene of interest and plasmid are cut with a restriction enzyme to yield single-stranded complementary overhangs. The DNA is mixed and the gene binds to the plasmid to yield recombinant DNA. The plasmid is then put into bacteria via transformation, and the cells are screened to find cells that contain the gene of interest. Transgenic bacteria and yeast are used in the pharmaceutical industry, in bioremediation, and the food industry. In the pharmaceutical industry, bacteria produce human drugs including insulin to treat diabetes and blood clotting factors to treat hemophilia. In bioremediation, genes are introduced into bacteria that allow them to break down petroleum after oil spills and other soil pollutants. In the food industry, bacteria produce the amino acid phenylalanine, which is needed for aspartame, which is an artificial sweetener, and yeasts are used to produce an enzyme that's used to curdle cheese to yield cottage cheese, for instance. Transgenic plants are generated by inserting the gene of interest into the T plasmid from the bacterium Agrobacterium tumefaciens, which naturally infects plants, causing a tumor-like growth, hence the species name tumefaciens, which kind of sounds like the word tumor. This bacteria is used since it readily enters plant cells. However, we don't want to make the plant sick, so the tumor-causing genes in the T plasmid are replaced with the gene of interest. The bacteria containing the recombinant T plasmid is then mixed with plant cells. The bacteria enter the plant cells where the plasmid inserts into the plant chromosome, and therefore the gene of interest inserts into the plant chromosome as well. The plants are then bred to yield homozygous individuals that all express the trait of interest. A gene of interest is put into the T plasmid to generate a recombinant or co-integrant plasmid, which is introduced into a plant cell where the gene incorporates into the plant cell's chromosomes. The cells divide to yield a small plant or plantlet, which grows into a transgenic plant expressing the gene of interest and therefore having the desired trait. Transgenic plants are modified to improve resistance to herbicides. Herbicides are chemicals that kill weeds. Weeds, of course, are plants, so very often herbicides not only damage weeds, but they damage the crop plants as well. Therefore, by improving resistance to herbicides, the weeds get killed, while the crop plants do not. Plants are also modified to improve resistance to insects. Insects feed on plants, which can kill the plants. In this case, the Bt gene from the bacterium Bacillus thuringiensis is introduced into the plants, and this gene encodes a protein that damages the digestive tract of insects. So when insects feed on these plants, they die, and this makes the plants naturally resistant to being eaten by these insects. The benefit to the Bt gene is that farmers need to use less pesticides, which are toxic chemicals that they spray on crop plants to kill insects. Transgenic plants are also modified to improve nutritional value. An example of this is golden rice, which contains added genes from the petunia plant and bacteria that enables the rice plants to synthesize beta-carotene. Beta-carotene is the precursor for vitamin A, so when the rice is eaten, the body converts beta-carotene into the vitamin A. Orange vegetables like carrots and squash contain high levels of beta-carotene. While these vegetables might be common in the United States, they are not common in Asian countries. In Asian countries, there was a high incidence of blindness from vitamin A deficiency because vitamin A is necessary to keep our eyes healthy. Therefore, golden rice was introduced into Asian countries, and since then, there's been a decrease in the number of cases of blindness from vitamin A deficiency. Transgenic plants are also generated to make them more desirable. For instance, to remove seeds, improve fruit color, or to delay ripening. For instance, there's genes that have been introduced to tomatoes that allow them to remain in the refrigerator for longer, and they don't spoil as quickly. Compare the white rice on the left to the golden rice on the right. The golden color comes from the beta-carotene. Transgenic animals are generated by injecting a fertilized egg with the gene of interest. This egg is then implanted into a surrogate mother, where the animal develops and is born. Because the fertilized egg divides via mitosis to generate the offspring, every cell in the offspring's body will contain the same DNA, including the gene of interest. Therefore, the offspring will express this new gene and have the desired trait associated with it. Uses for transgenic animals include the pet industry, research, the pharmaceutical industry, and the agriculture industry. In the pet industry, if you've ever heard of glowfish, these are genetically modified fish that contain a gene that gives them a fluorescent color. In research, human disease genes have been introduced to mice that leads to mice developing human diseases. This allows researchers to study human disease progression and also different treatments to try to help treat the disease. In the pharmaceutical industry, genes are expressed in livestock to produce human medicines that are secreted in milk. Since they're secreted in milk, it's easy to get the human medicines out of the livestock as they're purified from the milk after milking the animals. In the agriculture industry, animals have been generated to give leaner cuts of meat or to give quicker growth rate. Glowfish contain a gene that leads to their bright fluorescent colors. Their beauty makes them a popular aquarium fish. The DNA of the salmon on the top has been genetically engineered to produce growth hormones all the time for its entire life. Notice the massive size difference between this fish and the non-engineered fish below, which are the same age. This allows fish farmers to earn more money by selling larger fish in a shorter time period. People who eat this fish, however, will be consuming extra growth hormones. There is much controversy over the safety of this in humans. There are ethical concerns regarding GMOs. First of all, are foods from GMOs dangerous to eat? Well, despite the controversy involving extra growth hormones, for instance, the Food and Drug Administration has stated that there's no major safety threat that's been detected. However, they may cause allergic reactions. An example in the 1990s was when a gene from Brazil nuts was inserted into soybeans to improve the balance of amino acids in soybean proteins. Keep in mind that nuts and soybeans are good sources of proteins for vegetarians, so by combining the amino acids from both of these food sources, soybeans became a more complete protein. The problem is individuals with nut allergies that consumed these genetically modified soybeans had allergic reactions. Another concern is whether GMOs are hazardous to the environment. Keep in mind that modified plants may exchange pollen with wild plants to alter them. In the case of the Bt gene that kills insects that feed on the plants, this could lead to a decrease in insect populations if they have no wild plants that they can safely eat. If insect populations decrease, this could also lead to a decrease of all of the animals that feed on insects. We now start discussing different applications of DNA technology, including DNA sequencing, which determines the order of nucleotides in DNA. An automated version of the Sanger method is used. However, you don't need to know the details of how this procedure works. Overall, though, it generates different size fragments of DNA that can be visualized using gel electrophoresis, which separates DNA fragments by size. We'll discuss the specifics of this procedure coming up. Frederick Sanger's dideoxy chain termination method of DNA sequencing is illustrated here. Using nucleotides with special structures called dideoxynucleotides, DNA fragments can be terminated at different points, generating different size fragments which can be separated based on their lengths. These different size fragments can be read to give the sequencing results on the right. Gel electrophoresis is used during DNA sequencing and in other applications to separate DNA fragments according to size. The procedure is as follows. DNA samples are run through a gel-like material called agarose, which kind of has the consistency of Jell-O. The agarose with the DNA is put in a chamber that has a positive electrode on one end and a negative electrode on the other end. DNA is negatively charged, so it'll migrate towards the positive electrode when the current is turned on. Keep in mind that when it comes to charges, opposites attract. Smaller DNA fragments migrate more quickly than larger fragments to give distinct bands on the gel corresponding to size. The gel is then stained with a dye that binds to the DNA to allow visualization of the bands of DNA on the gel. Shown here are DNA fragments from several samples run on a gel, stained with a fluorescent dye, and viewed under ultraviolet light. The fragments run towards the positive electrode, with the ones at the bottom of the gel being the smallest. DNA sequencing has many applications, including the Human Genome Project, which was completed in 2003. During that project, all 24 different human chromosomes were sequenced to identify many characteristics. Keep in mind that there's 22 autosomes, plus an X and Y chromosome that needed to be sequenced. Characteristics of chromosomes, such as the overall organization, were identified. This includes which genes are included on certain chromosomes, as well as the order of the genes on those chromosomes. New genes with unknown functions were identified, and it's being worked on to try to figure out what the functions of those genes are. Genes associated with diseases were also identified, which is important in genetic counseling, which assesses the risk factor of an individual with a certain gene developing a genetic disorder. It's also important in trying to treat genetic diseases. Polymerase chain reaction is another DNA technology method that replicates a DNA molecule to yield many copies. It's essentially DNA replication in a tube using the DNA polymerase enzyme, which is the natural enzyme utilized during DNA replication in our cells, and this allows billions of DNA molecules to be produced from one starting molecule. This is because each cycle of PCR yields twice the number of DNA molecules. While the process may begin slowly, keep in mind that as the number of DNA molecules becomes large, each doubling really increases the number. For instance, once 100,000 molecules are generated, doubling then yields 200,000 molecules, then 400,000, then 800,000, etc. PCR yields enough DNA to work with in the lab for sequencing, cloning, or making transgenic organisms. An important application of PCR is for the use of DNA evidence in forensics. For instance, if a single speck of blood is recovered from a crime scene, it will contain very little DNA, but PCR will yield enough DNA for sequencing to take place to determine if the blood came from the victim or from a potential suspect. Polymerase chain reaction, or PCR, is used to yield many copies of a specific sequence of DNA. There are 3 steps that are used to separate the parental DNA strands and synthesize daughter DNA strands to yield 2 molecules from one. Each subsequent cycle then doubles the number of molecules. You do not need to know the steps or descriptions of these 3 steps, however. Short tandem repeats are important in generating DNA profiles, which we will discuss on the next slide. Human DNA contains many short repeated sequences called short tandem repeats. These are short sequences of DNA that are repeated over and over again. In the figure below, in the top strand of DNA, for instance, you see the sequence TCAT repeated 6 times. Short tandem repeats are found at specific locations on specific chromosomes in humans. Different people have different numbers of repeat sequences that can be used to identify an individual. For instance, on the previous slide, we saw the repeat TCAT repeated 6 times. While one person may have this repeat 6 times, another person may have it 2 times, while another person may have it 8 times, etc. Every person, aside from identical siblings who have the same exact DNA, has a unique pattern of STRs, which is known as a DNA profile. DNA profiles can be used in forensics to identify a victim of a crime or a potential suspect who committed the crime. DNA profiling is also used after natural disasters or terrorist attacks to identify victims. For example, after Hurricane Katrina in Louisiana and the 9/11 terrorist attacks, DNA profiles were used to identify the remains of victims.